Alright everybody, welcome back to another week of lectures. This week we get into the fun stuff again, so we're back into taxonomy and dissections. And going through taxonomy, this one's going to be kind of a doozy of a lecture. We're going through the last, or the major three groups that we'll talk about, so Echinodermata, Hemichordata, and then chordates. And then there's lots of taxonomy within those groups that we'll cover as well. So these three groups are all connected by the same characteristic of being a deuterostome. So a deuterostome, if you remember back to one of the first lectures that I gave, the difference between a deuterostome and a protostome. A deuterostome has the first opening of that blastula. That opening forms the mouth, or I'm sorry, the anus in a deuterostome, and then in a protostome, that first opening becomes the mouth. So everything that we've talked about from here on out for the rest of the semester is a deuterostome. We don't cover any more protostomes, so you can kind of forget that term for now. So here's a pretty good phylogenetic tree of everything that we'll try to cover from here on out. So these first two groups, Echinodermata and Hemichordata, are part of a larger clade called Ambulocaria. And so those two groups we'll be talking about first. Those two groups are sister groups to each other, even though they look really nothing alike. Everything else, Urchins, Cephalochordata, and then the chordates, or vertebrates, show some extreme diversity, and I'll go through that diversity in another lecture in, I believe, 2 weeks or so. So that first phylum that I mentioned, the phylum Echinodermata, is extremely unique in that it has secondarily developed a radial symmetry. So if you look back at that phylogenetic tree, you'll notice that it's sister groups to the hemichordates, and so the hemichordates display bilateral symmetry. They have that variform shape, so they look like a worm. But now you have these Echinodermatas that have split off from Hemichordata. They've branched off from that group, and they're displaying radial symmetry. One unique aspect is that the larvae of this phylum actually look just like hemichordates. They display this bilateral symmetry, and so we call this bipennarian larvae. There's 5 classes that we'll go through, and I'll take my time going through each one separately. So you have crinoids, Asperoidea, Ophiroidea, Echinoidea, and then Holothoroidea. So this is the class Crinoidea. These are the sea lilies, feather stars. They're an extremely primitive group. So many times, or most of the species that have been described for this group are actually fossilized. In fact, if you go out to any of the streams around here, you can find these fossils very easily. I think they're called Indian pennies around here. They just look like these small discs, and so those are part of this stalk. So we pull up a pointer. So those small discs that you can find in every stream around here are part of this structure right here. So this is the stalk, and this structure is similar to that of the Hydra or Hydrozoan that we saw earlier this semester, where there's 3 basic structures. There's a stalk. Instead of having a basal disc, it's called a calyx, which is right here at the base, and then they have these arms, or this fan-like structure, which then brings food closer to a mouth at the center of those arms. So every single organism that we talk about inside the class Echinodermata, such as like the Cstars or brittle stars, they'll have this calyx, and so this is just this central disc with basically a mouth on it. So this is the next class. This is the class Ophiroidea. These are the brittle stars or basket stars, and so they have these extremely long legs that you can see right here in this picture off to the side. So each of these legs is extremely long compared to the body itself. Those narrow legs are used to basically move this organism around along a substrate. So this is a fairly diverse group of organisms. There's currently 2,064 species that have been described. I believe every single one of them is marine. I believe most of them are actually from the Pacific Ocean. All right, so I mentioned very briefly that the highest concentration of the class Ferodia is in the Pacific Ocean. And so here's a really neat map from a paper that I'll post on Blackboard just for your own I don't know, benefit. I think it's a neat paper because it goes to the diversity of basically every species, all 2,064 species that we know of. But here you can see the diversity of this class. And so, as I mentioned earlier, the highest diversity is found in the Pacific Ocean, specifically around the coast, around Indonesia. So these organisms are found in entirely marine environments, and then more specifically, they prefer warmer environments. So right through here, you have an extreme diversity of habitat, marine habitat available. So you have these shallow areas closer to islands, such as the Indonesian islands, Philippines, Malaysia, stuff like that. But they have also these abyssal and hadal zones in each of these trenches. So right around here, you have Marianas Trench, you have, what's the other one? I forget what the other couple trenches are that are around here. But here you have an extremely deep area. So you have basically the two extremes in terms of depth. You have extremely shallow areas, and then you have extremely deep areas. And then everything in between in terms of habitat available for these organisms right through here. So lots of speciation has occurred in the Pacific Ocean for these organisms. As you can see, most of the colder areas, such as the Arctic, North Atlantic, North Pacific, and the Antarctic, are fairly non-diverse. Even the East Pacific Ocean. So if you think about water currents, warm water is flowing up from the equator from this Indo-Pacific region that I just mentioned. It's flowing up to the North Pacific. So the North Pacific is fairly warm on this west coast of, I guess technically the east coast of Russia, right here. But as it nears the Arctic Ocean, as it's closer to that pole, it cools down a lot. And so by the time it's coming through here, it's extremely cold water. So if you think about just the Pacific Ocean as like a big circle that's cycling water, as it arrives past California and past Mexico, it's fairly cold water that's coming down from the north. So this is why the East Pacific is fairly low in diversity compared to the Indo-Pacific on the other side. So neat little bit of biogeography for you all. And so that same paper that has this map published also has a chart on the number of described species. And so they were trying to make a species curve. So they plotted the number of species described per year. And so you can see, if you read that paper, which I highly suggest you do, you can see that there's still an upward trend. So if you want to join some marine biologists and study some bristle stars, you have a lot of room to work with. You can travel to some really neat places in Indo-Pacific or Pacific in general, and you might be able to describe a new species. So it's a neat group of organisms. Thought I'd toss in a little bit of biogeography for you all. So this third class that we'll go over is the class Echinodermata. So these are the sea urchins, sand dollars, sea biscuits. So this is a really neat group. They have this this exoskeleton, this calcium carbonate exoskeleton. It's covered in lots and lots of spines. So you can see here we have a sea urchin. Those are probably the most noticeable spines. But if you get a live sand dollar, that will have spines, very fine spines, along this entire exoskeleton. But once they're dry, and once you go to see them at a beach or like a store at a beach, all those spines have been removed, so you just get that calcium carbonate exoskeleton. So the big difference between this class and the other classes in Echinodermata is that these organisms don't have any arms. So if you compare these guys to the Cstars, Ferodia, Ferodia, or crinoids, they don't have any appendages that they can use to move food to their mouth. The sea urchin, it's kind of a shame that we don't have a saltwater tank anymore, just because last semester and the previous semester I was able to show people these sea urchins were using those spines and then their water vascular system to move those spines, bringing pieces of food closer to their mouth, which is on the underside of their body. So if you were to flip this sea urchin over, you'd see a mouth basically centrally located on the ventral side, so on the bottom. Within that mouth, there's what's called an Aristotles Lantern, which is this structure right here. I probably should have shown this picture earlier. So right here you have this Aristotles Lantern, and so this has muscle attachments that stretch out to this exoskeleton right here. And so on the underside of this Aristotles Lantern, there are several beaks. They kind of act like teeth, and they just start scraping away algae or tearing apart whatever pieces of food, or kelp or whatever. So that's the basic anatomy or body plan of the class Echinodermata. They're fairly simple organisms, but they've lasted for 600 some million years, an extremely long time. It's a fairly diverse group of organisms, so there's been 950 described species. I'm sure that number will probably jump up when more people take interest in this group. So the fourth group, or fourth class, within Echinodermata is the class Ferodia, and so these are the sea cucumbers. Just like the class Echinodermata that you saw last slide, these organisms don't have any arms. They just kind of look like this really fleshy, I don't know, cucumber, the best way to describe it, that you can find in the ocean. So these guys are very closely related to class Echinodermata that you saw on the last slide, but their spines have actually been they've shrunk, and so they're almost microscopic, and then they're embedded inside this leathery epidermis on the outside. So these guys are filter feeders. They just kind of move around on the substrate, and then they just start filtering the water in front of them, removing any organic material from that water. At least some of them are detritivores, so they eat decaying matter, and I believe another group within this class are going to be burrowing and eating, basically almost like a worm, deposit feeding. But they're a neat group. There's a couple videos on YouTube where you can see people picking these sea cucumbers up, and then they display a fairly unique defense mechanism. So they'll just basically throw up their intestines in order to clog up the gills of a fish or just discourage any potential predators. So those intestines will regenerate or regrow over time, but it's a fairly long process. So this organism can't just keep spewing out intestines. So it's a last resort defense mechanism for this group. This group's fairly diverse. It has about 1,700 species described. In fact, I think it's lower than that. I think it's 700 or 1,717 is the exact number. So the main group that you all probably recognize from Echinodermata are the Cstars, and so these Cstars are found in the class Ferodia, and so this is the typical body plan that one imagines with radial symmetry. So you have these 5 arms surrounding a central disc. So you can consider that a pentamerous symmetry or radial symmetry. So the central disc is right here, and then they have these 5 arms on either side. And so one of the things we'll be doing in lab this week is dissecting a Cstar. We'll be going through the water vascular system, starting from the madreporite down through that stone canal to the ring canal, down through all the tube feet. So I'll go over all that stuff in much more detail in lab, but just so you have a heads up on what we're doing. They do have some pretty neat regenerative abilities. So this is very similar to the intestines of sea cucumber that regrow over time. These Cstars can regrow limbs or an entire body from a limb. So this is if you take a Cstar and you chop off a limb, you'll have another Cstar starting to grow from that limb, and then that Cstar that originally lost the limb will grow another arm from there. So this is another fairly diverse group with roughly around 1,800 described species. So that's it for the phylum Echinodermata. We have tons of specimens, and so I've been setting them out on every single table, at least a representative of each group that we've talked about. So you all should have some ability to get familiar with specimens for the lab practical, or final lab practical. So the next phylum that I mentioned earlier on was the phylum Hemichordata, and so these are the acorn worms, and these are very, very primitive organisms. These guys are filter feeders, and they're also deposit feeders. So once again, they're just burrowing through the sediment within the ocean floor. So these guys are entirely marine, and they have almost all the characteristics of a chordate. So they have what's called dorsal hollow nerve cord, pharyngeal gill slits, they have a postanal tail, but they don't have a notochord. So this group was originally considered part of the phylum Chordata until it was determined that they don't actually have this one synapomorphy, the presence of a notochord. So they were kicked out from the phylum Chordata and given the name Hemichordata. So we have one specimen of Hemichordata right here in this small vial. It's pretty recognizable compared to every other specimen that we have. So it's, yeah. But yeah, it's a neat group. They have a proboscis that they use kind of to poke around the soil with. These small blue lines show the pharyngeal gill slits, so they're basically pumping food into their mouth through that proboscis, and then they're filtering food and then passing that detritus outside those pharyngeal gill slits right around here. What else? Yeah, they're a fairly non-diverse group. There's only about 130 described species, and they're all marine. So that's the phylum Hemichordata, and that brings us to the phylum Chordata. So these are the chordates. So we're going to go through the 3 major groups. So you have the vertebrates, Cephalochordata, and then Urchins, and then I'll go through diversity of chordates in another lecture in a couple weeks. So there are 5 main characteristics that define a member of the phylum Chordata. And so the first is that they have a notochord. And so this is a rigid structure that is providing some sort of support for the next characteristic, which is a dorsal hollow nerve cord. So you have this nerve cord that runs down the length of the body, basically controlling whatever nervous impulses are being sent from the brain at the very front. So you have cephalization of these organisms. You have that brain in this, the very front or the anterior end. Any nervous impulses are being sent down this nerve cord, down the length of the body. And so that nerve cord has to be dorsal, so located along the top of the body, and then centrally located. It has, or the next defining characteristic is having pharyngeal arches. And so these structures are right here that are labeled gill slits. Those are the pharyngeal gill arches. And so they are these openings into the body that act as respiratory structures early on. They can be modified into thyroid glands. I'm sorry, that's the endostyle. They can be modified into voice boxes and stuff like that. The other characteristic that I just mentioned, so I'm going to skip ahead one, is endostyle. And so this is modified into a thyroid gland in most mammals, but in very primitive chordates like Cephalachordata or Urchins, they have what's called an endostyle. And so this is producing hormones or regulatory chemicals for this organism. So the characteristic that I skipped over is number 4, and so this is a postanal tail. And so this is pretty self-explanatory. You have this fairly muscular body, and it's swimming throughout the whatever solution, water, fresh water, whatever. And so it then has this fairly muscular tail that it's using to swim, and then its intestinal system or digestive tract ends before the end of that that body. So this is separating this group of chordates basically from all the other worms that we've talked about, or worm variform-looking organisms. So you have a mouth on one end, but you don't have an anus on the other end. You have basically this muscular tail instead. So I like to show these pictures just because it shows, well, at different stages in development of organisms, we all kind of have the same 5 characteristics that define this phylum. So you have a fish, frog, an alligator, a chicken, and a guinea pig pictured here. And so you can see they all have this postanal tail right here, here, and here. What's maybe not so noticeable are the pharyngeal gill arches. You can't really see an endostyle. Maybe you can see a little bit of the notochord. So here on that guinea pig, you can see this this central that vertebrae that's developing right there. You can see the same thing in the chicken. The alligator is not translucent, but in the frog and fish, you can definitely see this this rigid structure right here that it's using to basically protect that dorsal hollow nerve cord, and then also give it some structure or rigidity inside of its postanal tail. So this picture at the top right is a SEM image of a human embryo at 25 weeks old, or I think 3 weeks old, not 25 weeks old. I don't know why I said that. So this is an extremely young embryo. It's just developing some of the characteristics. And so what you can see here are these pharyngeal gill arches. These are true gill arches, so these are remnants of, well, past evolution. So these gill arches will condense into our voice box. Some will travel upwards and form the inner ear bones. So even though we don't have the gill arches displayed prominently in our adult stage, during our larval stage, when we're forming in the womb, we have these pharyngeal gill arches, and they are modified, or they develop into other structures that we use every day. We even have a postanal tail very early on during our development, but after a while, I forget what gene encodes it, but after a while, there's a regulatory gene that basically stops the production of that tail, and so that's why we don't walk around with a tail. In almost all vertebrates, that endostyle is modified into the thyroid gland. So in humans, we have thyroids, and those regulate the chemicals or hormones inside of our body. All right, so the first subphylum within the phylum Chordata that we'll talk about is the subphylum Meteorochordata. And so these are unique in the fact that only the larvae are displaying all 5 characteristics. The adults are only displaying 3 of them. So the adults have a notochord, and a nerve cord, but only the larvae are displaying all 5 characteristics. So they have a notochord, postanal tail, nerve cord, pharyngeal gill slits, and endostyle. So the larvae, I don't have a picture up here, but the larvae are free living. They're swimming throughout the water column. And then as they develop into adults, they'll settle onto a substrate, whether it be a rock or whatever. And then they just kind of form almost like a barnacle-like structure where they're sessile for their entire adult lives, and they're just filter feeding the rest of or that salt water. So they have an incurrent and excurrent siphon. So water is flowing down that incurrent siphon past pharyngeal gill slits, which have been modified in the adult stage to kind of act like a filter and start moving food towards the stomach. So that incoming water travels throughout that central tube and then exits out the excurrent siphon. So right here you have the anus. So this is the end of the digestive system, but you still have this remnant of a postanal tail. So the structure right here extends past the anus of this organism. So here's another couple pictures. Once again, I really should have included a picture of the larvae just so you can get an idea of what they look like. But here's a diagram. So you can see they have this notochord, this rigid structure that's running right here that's giving some sort of protection to the dorsal hollow nerve cord, which is right on top. The endostyle is labeled here. They have a postanal tail. They didn't show the anus on here, but it would be right around here. Right after the stomach, so food's traveling right here, takes this kind of curve downwards and exits out the body. So these 2 pictures here and here are once again of the adults. They're sessile and they're only showing a postanal tail and an endostyle. So they don't have that notochord or dorsal hollow nerve cord being displayed. So this group is unique in that aspect. So the second subphylum within this phylum Chordata is the subphylum Cephalochordata, and so these are the lancelets or the Amphioxus, and so these are a really neat group of organisms. We have a couple jars. They really just look like very thin muscular worms. And so these are filter feeders. They're entirely marine. If you look at the head of one of these lancelets, you can see it has basically this fan-like structure that it's using to just sift through sand or whatever organic material is coming in with the surf. So these organisms will sit kind of buried in the substrate, like you see here. So they're buried in the substrate up to this point, and then they sit with those heads out that fan, and they just kind of scoop this organic matter into their mouth. So these are a neat group, very non-diverse, only 30 described species. And so these split off from the rest of the chordates, from Meteorochordata and Chordata, roughly 600 million years ago. So this is an extremely primitive group, very, very derived. But it has all these characteristics, those 5 characteristics of a chordate present in both the larval and adult stages. So Urchins, it's only found in the larval stages. Cephalochordata, it's found in both larvae and adults. And then the next subphylum, Vertebrata, you should be a little bit more familiar with already. Excuse me. So here's a picture or a diagram of a lancelet. So you can see it has this dorsal hollow nerve cord in light blue, runs down the length of the body from the head. So there would be a brain right around here. Basically, this dorsal hollow nerve cord, it folds around itself and then forms a brain. They have this notochord that runs down the length of the body. I don't know if you can see it in the specimens. Maybe I'll dissect one and open one up for you all. But you should be able to see a notochord here. You can't really see it in either of these structures or pictures. These organisms do have pharyngeal gill slits, so once again they have these structures that they use to basically prop open their body and allow them to pump water through the mouth and then pass these gills on their body. They have the endostyle, which is this dark blue structure right here, and then they have a post-anal tail, so you can see the anus right here, and then they have this muscular structure just posterior to that. So one neat thing that you'll see next week are myomeres, and so these are really muscular bands that these organisms can use to basically move around through water or the substrate. And so these myomeres are also found in fish or lampreys. And so it gives this organism the ability to really undulate its body and swim through the water. So this brings us to the last subphylum that we'll talk about. And so this is the subphylum Vertebrata. I'm sure you all are very, very familiar with it, considering that we are in the subphylum Vertebrata. We have a vertebrae. So instead of having a notochord, our notochord has been replaced with this calcareous structure called a vertebral column. I don't have any examples close to me. But this is an extremely diverse group, something like 60,000 species. So this includes the fishes, amphibians, reptiles, birds, mammals, lampreys, hagfishes. It's extremely diverse. It represents a wide range of organisms. So, as I just mentioned, that vertebral column replaces the notochord. So this structure right here in this fish, this is the vertebral column. This is protecting that that dorsal hollow nerve cord. So the brain of this fish is located right here, and then it runs the nerve cord down the length of the body, so that can control the movements of those myomeres or those muscles. And then there's this vertebral column that it's using to protect that spine. So in 2 weeks, we'll start our pig dissection. So I was asking a couple of the sections what their opinion was on starting a pig dissection early. So when I begin the chordate taxonomy lectures, we're just going to very briefly skim through the specimens, and then we're going to start with dissecting a pig. So the reason why we're starting 2 weeks early, or a week early, for the pig dissection is that it's an extremely intense and long dissection. It's a very complex organism. So to go through every structure in 50 minutes or whatever that we have allotted is almost impossible. So spreading that out over 2 weeks will give you all some more time to familiarize yourselves with the specimen. So yeah, next week, so not this week, this week we're dissecting Cstars and fishes, but next week we have our behavior lab, and so this is a fun one. We deal with live mice, so I'll talk about that when we get there. Make sure you're wearing closed-toed shoes to lab this week. As I just mentioned, we're doing dissections again, and I'll see you all Wednesday or Thursday.